Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 13th November 2020. We are into the revision mode and we are revising all important chapter number 8, cell structure and functions. As we have been discussing, we realized and understood that cell is the structural and functional component of any living organism, and the cell having inside so many parts, we call it cell organelles, play crucial role in building up of the tissues. And the very cell will be able to be alive because of the presence of these organelles. Each organelle performs its designated function, and in the presence and continuance of this function being done by these cell organelles, cell will flourish. As part of it, we were talking about the functions and the structure of various organelles, and today we are going to talk about another important cell organelle called vacuoles. So vacuoles are non-cytoplasmic areas present inside the cytoplasm, formed by expansion and pinching off from endoplasmic reticulum. So these are the byproducts of ER basically. There are varieties of vacuoles. In fact, sap vacuoles separated from cytoplasm by tonoplast. Contractile vacuoles take part in osmoregulation and excretion. Food vacuoles are formed by fusion of phagosome and lysosome, and they help in digestion of food. And the food vacuoles having digestive enzymes help in digestion of the nutrients. So vacuoles are membrane bound, space bound cytoplasm containing water and sap and excretory products. They are bound by single membrane. They form contractile vacuoles and food vacuoles in many organisms. And then comes the very important, very powerful cell organelle. These are called battery of the cell, mitochondria, powerhouses of the cell, mitochondria. They're called so because of the ability to store the ATP molecules. Just like a battery stores energy, mitochondria stores ATP molecules inside it. Hence, they are called powerhouses of the cell. They are called battery of the cell. It's a double membrane. You got outer membrane and inner membrane. So two layers are there, and as you are seeing in the diagram, the inner membrane is having foldings internally, and these foldings form finger-like structures. These finger-like structures are called cristae, and the cristae will be floating in a fluid called matrix. So in the cristae, which are finger-like structures like this, there will be what we call F1 particles. So these F1 particles are very crucial in the storage of ATP molecules. So F1 particles are the specific ones inside the mitochondria that play a crucial role in storage of energy, and these F1 particles are present in cristae. So mitochondria is a double membrane bound structure with outer membrane and inner membrane, and inner membrane is entering into the lumen and forms compartments, finger-like ones. The inner membrane forms number of foldings. These finger-like foldings are called cristae, and there will be matrix. The two membranes have their own specific enzymes. That is the uniqueness of mitochondria. Mitochondria are sites for aerobic respiration, as I said. They produce cellular energy. This is ATPs are synthesized here and form ATP. So they are called powerhouses of the cell. Many times in UPSC, NDA, this is being asked. The matrix of mitochondria also contains circular DNA molecules. This is the uniqueness again. The organism as such will have DNA in every cell inside the nucleus, but in addition to the presence of DNA molecules in the nucleus of every cell, the mitochondria also will have its own DNA molecule. That's the thing. And also a few RNA molecules, ribosomes, and components of protein synthesis. Hence, mitochondria is all important. And thanks to the scientist who discovered this, Kolliker in 1850 way back, and the outer membrane which is smooth and has porine proteins, the proteins called porins are present that form channels, and the inner mitochondrial membrane which is semi-permeable and regulates the passage of materials in and out of mitochondria. It usually produces numerous infolds called cristae, which provide the inner mitochondrial space into many compartments. The cristae and the inner face of the inner membrane is studded with the particles called oxysomes. Remember, oxysomes. And the other name for this oxysomes is F0, F1 particles. F0, F1 particles. The oxysome having the F0, F1 complex represents ATP or ATPase, adenosine triphosphatase. And this ATP synthetase enzyme is very essential for the formation of ATP. Hence, mitochondria plays very crucial role. Next comes plastids. Again, these are very, very important. These are called kitchen of the cell because they synthesize and store the food that is required. Hence, plastids are also as important as the mitochondria. And the plastids are of three types. There are green ones. Green plastids are called chloroplasts, and they contain chlorophyll. And chlorophyll has got varieties of chlorophylls ranging from A to F. And chromoplasts, these are having keratin and xanthophyll. These are having colors like red color, brown, yellow, sky blue, like that. And then leucoplasts, these are colorless plastids, and they have the important function of storage of starch. Hence, they are godowns, we can say. They are godowns of the plant cell. Chloroplast contains chlorophyll, and chlorophyll that traps the solar energy. Recollect that light reaction, Hillman reaction, remember? And 740 to 780 nanometers that we discussed in photosynthesis chapter. And the chromoplast provides yellow, orange, red colors to different parts of the plants. And leucoplasts are the colorless that store food. Amyloplasts, that's carbohydrates. Eleoplasts, oils, and alluroplasts, that are proteins. Chloroplasts are double membrane structures, just like mitochondria. The space is limited by inner membrane called stroma, and the thylakoids are present in stroma as stacks like the piles of coins called grana. Now, the transfer section TS of plastids reveal that any plastid is made up of two membranes, as you are seeing, outer membrane, inner membrane, and inside you have got bag-like structures. These are called thylakoids, and a stack of thylakoids, just like if you take 1 rupee coins and arrange 1 rupee coins one above the other, say 10 to 20 rupee coins you arrange in a row, that will be a granum. So the thylakoid will be like a bag, and inside it a lot of enzymes will be there. And this granum having a stack of thylakoids will be more in numbers inside the stroma. And the stroma, inside the stroma, each granum will be interconnected by small tubules. They are called stroma lamellae. So stroma contains enzymes for synthesis of proteins and carbohydrates. And double-stranded circular DNA and ribosomes are also present in stroma. Eukaryotic cells have ATS ribosomes. They have granular structure with 2 subunits. So this is how plastids are largest cell organelles. These are involved in the formation and storage of soluble organic compounds. And chloroplasts are greenish plasmids which possess photosynthetic pigments. It's bounded by 2 unit membranes separated by space called periplastidal space. Part inner to membrane is divided into 2 portions. One is grana, and the fraction consists of membranous or lamellar system made up of thylakoids, And the second one is stroma, is a proteinaceous complex having proteins, ribosomes, DNA, enzymes and lipids. And chloroplasts are semi-autonomous organelles. This is important. And leucoplast, on the other hand, colorless plastids found in storage organ away from sunlight. Chromoplasts are non-photosynthetic plastids and having variously colored except green. These store carotenoid pigments, and chloroplast is the center of photosynthesis. Recollect from your memory: light reaction and dark reaction, Hillman reaction and Blackman reaction. So you got the chloroplasts and mitochondria both together play a crucial role. Endosymbionts in eukaryotic cells. Both chloroplast and mitochondria are semi-autonomous organelles. Both are evolved by two unit membranes, outer smooth and permeable and inner unfolded and semi-permeable. Both have circular DNA, 3 types of RNA and 70 S type of ribosomes. Both are energy transducing organelles and form ATP by a similar mechanism. Both mitochondria and chloroplast occur only in eukaryotes and are absent in prokaryotes. So that's how the mitochondria as well as the plastids, particularly chloroplast, play a crucial role, and they are most important organelles for the plants. So let's continue our discussion in our next session about other organelles. Until then, keep practicing, draw the diagrams, label it, and I want you to post it in our WhatsApp group. The other day I have told you, but you have not obeyed my order. Now this time I will be very serious. If there is no response from you, I think I have to resort to some other means. Otherwise, how do I know that what I am teaching to you, you are able to follow, you are able to understand? Because already it is revision. Once videos are already over, you have seen the videos, and this is only revision. And I am sure you must be having a separate notes, revision notes, as I told you. And if not done, please start now, today only. And I want you to draw diagrams of concerned chapter, label it, and below the labeled diagram I want you to write the importance of